results and used for a group analysis. So why would you want to be in this space compared to like a traditional nifty M&I transform space? Well, there's the benefits of surface analysis, obviously. So there's surface smoothing, so not smoothing CSF and white matter signal into your gray matter signal. There's also surface visualization of your data, which is just a lot more easy and straightforward to do. And we all like to have those really pretty surface plots in our things. And it also allows for better cortical registration across subjects, which is a big key for me. And I'm very interested in looking into the implications of that. But also something you get out of and also potentially more multimodal approaches by just having cortical thickness and resting state data and other formats actually in the same file format. The other thing that it does is it does this without leaving data from the subcortex behind. So a lot of other packages that have been put forward for working on cortical data do it in a way that basically throws out the subcortical data from your pipeline analysis steps, and it's really hard to reintegrate subcortical data into your analysis afterwards. Okay. So the reason why not to use this is because so far it's been kind of difficult. It's actually taken a while for me to work through the code and figure out how to do this properly. So I've been kind of recording everything I've been learning in this GitHub repo I call Siftify and packaging it into smaller tools. So I've been building tools, some of them are conversion tools, so I now have a tool that will take any FreeSurfer output and convert it into structural SIFT data, and use the FreeSurfer output to get the surfaces and the subcortical segmentations. And I also have another script which will work after that, which will take any preprocessed functional file already still in native space and transform it into this surface and subcortically mapped format. I also have another tool which is, I call it Siftify and Nifty, and it's for projecting any data, basically to projecting any data with a fewer steps, which is more for projecting masks to the surface. So going from an M&I space volumetric mask to going to an M&I space to the SIFT format surface masks. And there's also some tools I built around QC, which is why I wanted to actually introduce this talk before our morning discussion of QC, which builds upon these packages in this file format. So one of the main things that I do this for is QC of the FreeSurfer pipeline. And so once you have it in this format, actually the Human Connectome project tools have really great kind of backbone snapshot and picture generalizing things that can be scripted upon. I've been building these formats, which basically recreate some of the FreeSurfer QC pages you've seen in the past. But some of the things that I added, which I wanted for QC, which don't exist in other packages, are these kind of all subject index page views, which I find really useful. So for every view that I create like a subject page view, which I think is what we're all used to when we're actually doing like manual quality assurance, like clicking on every subject and then deciding whether or not that subject is good, and then moving to the next subject. But we usually don't actually look at all of the subjects together at the same time, and I would argue this is a really useful thing because it's really, you can actually be much more precise about what your cutoff is for good and bad when you're looking at your entire population in the same slice, from the same slice view together. So I created these views so you could look at the say, surface reconstructed brains of basically your whole population and scroll down through a web page and pick out the ones that were good and which ones were bad. So it's hard to see on this slide a little bit, but most of these are good, but this guy in the middle is what I call an epic fail. Like, just FreeSurfer did not work for this person. I also took it a step further and created these visualization pages for resting state data or for functional connectivity once you get it into this file format. So I created this little tool that basically takes your output, drops a seed, creates the seed correlation image, takes pictures of that seed correlation image, so pretty surface reconstructions of that correlation image, and then just throws out all the intermediate files. So you can look across your entire population at all of their functional seed connectivity patterns And then you can look for really abnormal ones, so ones where there's probably a lot of noise in the data. There's probably like a bad functional scan to start. And you can and actually, by default, you use 3 standard seeds that usually have a normal pattern of functional connectivity. And I put it all in this pretty bootstrapped page. Oh, and then on top of that, I thought instead of looking at only functional connectivity, you can look at anything else. So you can actually give this essentially any kind of intermediate Nifty file at this point, and it will just create a visualization page for you to like look over what all of your subjects look like in like one view. So I thought that that was quite useful for QC, but moving forward with more discussions about quality assurance and issues that you have in multi-site and population data, we're going to move into our chairs, our last planned conference talk from Stephen Struther. I'll bring him forward. Oh, thank you. And we're running out of time, but do feel free to come forward or talk to me with any questions about this package and how it might be useful. Okay.
We should talk to you about your package. What is it, PGA? Yeah. Hopefully something's happening. Searching for source. Not necessarily a good sign. Okay. Well, my computer thinks I'm ready to go. Okay. Great. Can we wander around with this? Okay. So I'm going to talk about multi-center challenges in resting state fMRI and tell you about a problem I didn't know existed until about 6 months ago, and I still don't know how big a problem it is, so I'm interested in people's ideas. And I know you're all really excited about brains, and I am as well, but this is actually going to be about MR scanners and phantoms, because it turns out there's some challenges in certainly the temporal dynamics of some of these signals that you're looking at. That's just about me and the standard disclosure, because I have a company, so it's just easiest to put it up. So I like to think about neuroimaging pipelines, and as I mentioned yesterday, trying to make optimal decisions. And everything you do when you run an experiment on an MR scanner is subject to the idea of, did you make a good decision or not? And therefore it's subject, if you can work out how to do it, to quantitation, because that's what science is about, right? So your subject selection, your experimental design, your data acquisition, and this is the stuff I usually talk about. So the pre-processing, optimizing that, choice of data analysis, particularly in a machine learning environment, how you choose metrics to drive all that. But that's not what I'm going to talk about today. What I'm actually going to talk about is back down here, these first 2 stages, the experimental design and the data acquisition, and particularly how they influence multi-center effects. And when I set up this diagram, which you see here, I didn't actually realize that we needed to worry so much about the scanner effects, or at least not in the way I now understand. So the goals for a multi-center or a multi-MRI scanner study, where our aim is to get resting state fMRI acquisitions. What's the contribution of between session, within center scanner effects, so the scanner stability type issues, and between center scanner effects, so scanner models, etc. And how should these effects best be removed? So when we started on this exercise, I thought, well, people have been thinking about this for at least 10 years. I bet I can go out into the literature, I'll find a solution, and I knew a group in the US had been working on this for a while. We'll just run that through our stuff. We'll get the same results they get, and we'll just move on to more exciting and interesting problems. And unfortunately, that wasn't exactly what happened. So we're doing this in the context of the Ontario Brain Institute's integrated discovery programs, specifically the Ontario Neurodegeneration Research Initiative, in which I run the Neuroinformatics Core, and the Canadian Biomarker Integrated Network in Depression, in which I'm co-PI of the neuroimaging section. And the ONDRI there is about 15 centers for patient recruitment with 10 different scanning centers. And CAMBIND is something like 12 centers for patient recruitment. CAMBIND's actually right across Canada. ONDRI is just within Ontario, and CAMBIND just has 6 scanning centers. So we end up with about, I think it's 13 scanners in total, mainly in Ontario, but some out west. And we're just going to focus on the neuroimaging piece here. And so the first thing you do is you design an acquisition sequence, and this was done by Rob Bartha and Simon Duchenne and a whole pile of people. So there's a standard dementia imaging protocol called CDIP, which you can go look up on the web if you happen to be neurodegenerative scanning. This has been used in the Trans-Canadian CCNA, the Canadian Consortium in Neurodegeneration in Aging, and it's also being used in ONDRI and a number of other very large studies. So it's at least worth knowing about if you're interested in aging and neurodegeneration. You can see we have to deal with GE, Philips, and Siemens, and this is just the resting state sequence. There's a whole pile of other acquisitions I won't talk about. So the things that I just want you to note is it's a very standard, very conservative sequence, because we didn't want to push any of the scanners. There's no multiband or anything like that. So the TR by modern standards is actually relatively long at 2.4 seconds. It's voxel size of 3.5, 3.5 by 3.5. And there's 250 TRs, so it's a 10-minute total sequence. So it's a reasonably longish acquisition, but not presumably pushing the scanners really hard. So if you want to find out about scanner effects and get rid of all that annoying human stuff, which just makes it really hard to see scanner effects, the best thing to do is take away the human and replace them with something inert, unless you have an inert human available. Actually, I shouldn't joke, because there are actually a couple of papers scanning cadavers, but we couldn't get a cadaver, at least not across the 13 sites in Canada, and shipping one around seemed logistically to be a bit of a challenge. So it's actually much easier to look in the literature, and there's a study called the FBURN, the Functional Bioinformatics Research Network. This was the first big multi-center study in the US that focused on fMRI and tasks in schizophrenia. And they produced a whole lot of papers over the last decade. And one of the things they did was they designed this phantom on the left here that you probably can hardly see, but it's an acrylic sphere full of approximately tissue equivalent gel, and you can buy it. It's made in some garage somewhere in San Diego, I forget, by Carroll. Anyway, we bought a whole pile of them. They're less than $1,000 each. That's a scan of what they look like, and it's much easier to work out what the scanner effects on this uniform, nice piece of geometry than it is on that there. And in fact, one of the challenges is taking the things we're doing in the phantom and actually moving them across. So how do we actually detect the same effects in human data? Because you've got to somehow separate it out from all the things that heads do when they get into the scanner. So the FBURN guys came up, particularly Lee Friedman, Gary Glover, and Doug Grieve, came up with a pipeline that you can download from the web. There are 16 QA measures, and these have actually sort of migrated into various other QA packages that are around. So I think Cameron Craddock's package uses at least the full width half max resolution pieces and then adds stuff. I know there's a package at CAMH by Joseph Viviano, which uses some of this and then changes it. Russ Poldrack uses some of it and then changes it. So unfortunately, our quality assurance procedures are breeding in the same way that all our other analytic and methodology tools are. So now someone, and you know, I guess I'm possibly one of the people who's going to do it, has to work out whether these things are all the same. Are they different? Do some of them have new parameters that we should actually use and worry about? So we're in the middle of thinking about that. There are 16 here. I'm happy to talk about them later if you're interested, but I don't have time at the moment. I mean, they're the sort of standard things: signal to noise in space, signal to noise in time, drift, percent fluctuations, the forward half max measures of various sorts, which I'll talk quite a bit more about, and mean ghosting. Phantoms are really good for getting ghosting measures because they have a nice defined geometry, so you can find it and then move off and check to see whether there's extraneous signals sitting around outside, which is exactly what they do. In the OBI projects, we use these things, dashboards, just to display all these results. These are all the individual parameters, just as bar plots. Here's all our phantoms. So there's about 410 phantom scans we now have across all these scanners. There's 5 Gemens, 6 Siemens, and 2 Philips. It's scanned. The phantom's supposed to be scanned once a month at each site. If you ever embark on one of these, don't underestimate how hard it is to get remote people to do something for you, even when you're paying them. Because it's not really high on their local priority list typically. So we actually have a whole overhead, which is just to make sure these phantom scans actually get generated. So it may seem sort of trivial, but to get 2 years worth of phantom scans across 13 scanners turns out to be a whole infrastructure challenge, which I didn't appreciate. We can also look at this stuff longitudinally. So this happens to be the scanner at Baycrest. And you can see this sort of stuff going on, wandering in and out of ghosting, although it's within plus or minus 2 standard deviations, so maybe we wouldn't worry about it. So we started off with this idea that we had these 16 parameters, and we'd go ahead and we would put standard deviation bounds in the standard, you know, sort of univariate way, very simple-minded quality control. And it quickly became obvious that trying to track these 16 parameters across 13 scanners over time, I mean, you can say, yes, that went at outside 3 standard deviations at some particular point, but it really wasn't helping us gain any understanding about the relative performance of the scanners, which was one of the goals. So we decided to use my favorite friend, which came up yesterday a couple of times, and that's a principal component analysis. We threw it all into the mix. And we actually cleaned it first because we're also tracking the acquisition parameters, and sometimes the sites, for reasons that still escape me, manage to use completely different acquisitions than the one that we've agreed on and that they've agreed to run. And that happens on human scans as well, by the way. That's just another part of this whole social engineering piece when you do these multi-site studies. So we track that, so we removed everything that didn't come very close to our defined parameters. And then we threw all of these into a principal component analysis, and on the left here, we have the 2 components, PC1 and PC2, so the highest variance, second highest variance. These are the weights or the saliencies of each of the 16 parameters. And on the right, you have the distribution over the actual phantom scans themselves. And I have no idea if you can see, but you can actually track time in this because there are lines with arrowheads on them joining. So this is a scanner that sat over here for about a year and then went through some spontaneous conversion process and decided to wander over here. And that was being used all that time as a research scanner for all sorts of studies, including clinical trials. Let's see, let's go to the next one because you probably can't see that very well. Hopefully you can see this a bit better. So these are the saliencies or the weights on the individual variables. And you can see here that the first component is being driven by full-width half max in the x and y direction in each slice. And you get a minimum and a maximum value because it's a statistical measure the way they do it in FBIRN. They actually use a finite difference, a local finite difference measure. But basically, this is a resolution axis, okay? And the other axis is ghosting. You can see you've got mean bright ghost and mean ghost. So thinking that this is a resolution axis and this is a ghosting axis, this is one manufacturer's set of scanners. This is another manufacturer's set of scanners. Anyone want to guess? No? Yeah. This is Siemens. So I'm sorry for those of you with GEs, but at least in our experience, the Siemens scanners tend to be much more stable over time. Because remember, this variability in this horizontal direction means that their resolution is varying for some reason as a function of time. This is a mixture. This is GEs and a couple of Philips over here. That's the CAMH scanner there, if anyone's interested. It had a little hissy fit at some point. I don't quite know why. And that's also a GE at the top there. This is a scanner where you can see that had purely a ghosting problem. It stayed at exactly the same resolution, and that turned out to be about a 200 hertz change in the pixel bandwidth, which took quite a while to sort out. So we can now measure this stuff, we can see what's happening as a function of time, and we can go back and look at the actual full-width half max measures just to get a sense of quantity. So the full-width half max, the minimum value in all the Siemens is about 2.6 millimeters, and the full-width half max minimum value in the Gs is 3.9. That's just the way they do their standard case-based reconstructions. And this was first published in 2006 because they saw it in the FBIRN study when they were doing fMRI. So Bob Cox was talking to Gary Glover and Doug Grieve, and well, that's interesting, it's disappeared. Just a second, no, well, anyway, there's a standard package in AFNI which allows you to define a resolution. It goes in and measures it in the image that you're trying to smooth, and then it adapts the smoothing filter to try and hit that resolution. It'll run on both humans and phantoms, but there's some issues with the humans. So we ran that, because that's the standard literature approach to trying to fix this issue. And here we have our plot. So the percentages have dropped a bit, and they've become a little more equal. That's what you would hope to see. So you're flattening out the eigenspectrum, which means we're getting rid of some of the variance. It's actually rotated, which I guess I'll show you in a minute, so let's blow this up. So what's actually happened now is the max full-width half max has become opposed to the minimum full-width half max. So something about trying to equalize the resolution has actually pulled measurements within a single time series session per slice a long way apart, and they're now helping drive some of this structure. So basically there's sort of two rotated dimensions here. There's still a resolution dimension in that direction, and then there's something else going on involving ghosting and signal to noise and a range of other things in that direction. So this is what it actually looks like, and this is the scale. So in fact we have on average got, these are all the Gs and Philips here, and they're the Siemens there. So we've certainly brought them closer together. So it's doing, first order, it's doing what you would hope it would do. But remember, that is a resolution direction. It's creating all these outliers. So it's done something good and something not so good. So we then decided we'd better work out what the not so good piece was. And because I tend not to trust software that I wasn't involved in setting up in the first place, So we decided to use another way of measuring the resolution using the autocorrelation function, which is pretty easy to do using Fourier transforms. So this is on second order detrended time series. All the other stuff has also had detrending because you want to do these measures on the random noise field after you've removed sort of structure, temporal structure. And this just shows you, this is a relatively high resolution image. That's the autocorrelation function in X and Y. This is a lower one. You're essentially measuring the size of these blobs. So you can go ahead and do this on the 5,000 slices that we have in our standard resting state sequence. So that's 250 TRs, and we're only looking at the central 20 slices on the phantom to try and avoid some of the geometry issues because it's a sphere. And on the left here you have two well-behaving sessions, okay, giving standard stuff. So on the right here, you have a Siemens scanner where just one slice at 1 TR decided to have a little fit and just did something weird. At the bottom, we have a GE scanner where lots of slices decided to do weird things. So now the issue is, how do we quantitate that effect? This is one way. So this is the GE. So on these plots here, I'm plotting as a function of TRs along the horizontal axis, 250 of them, the measured full-width half max using the autocorrelation function across, at every one of these points, 20 slices, and I'm plotting the max value across the 20 slices, the average value, and the minimum value. So what you see is you go across the TR, and it's shown graphically down here where we put a threshold on this and then just put a black dot in the slice that goes above threshold. You can see the scanner goes in and out of some sort of stability which is being clearly seen in the full-width half max measure. And this turns out to be low spatial frequency non-uniformities. So what you can do with that thresholding is say all these slices with the black dots, let's just call those outliers and let's average them all together and look and see what the pattern looks like. And we're going to normalize it by the grand mean of every voxel across all TRs and slices that we collected, and that's shown here on the left. That's actually in percent, so that's only 0.2%, down to minus 1.2% variability. But here are all the non-outliers. That's all the slices in these places where the autocorrelation function seems okay. And you look at the huge difference in the size. That's 0.02%. So 0.02% is about what you should get with this phantom in a well-behaving scanner. But to my surprise, scanners appear to throw these weird instability, little fits of instability, somewhat asynchronously and randomly across slices. And GEs and Philips are a little more prone to do it based on our experience than Siemens, but virtually every scanner does it at some point. And the percentage changes are sufficient that if there's enough of them, they're really going to screw up, or potentially will screw up, resting state dynamics. They're not going to have much impact necessarily if you have a nice long time series on, for example, extracting the spatial default mode locations. And if you go look at all the reproducibility studies in the literature, an awful lot of them are about, you know, did I get all my default mode nodes that I expected to get with some reasonable reliability? Well, it turns out that's a pretty strong signal and it's pretty easy to get. If you're interested, as I am, in trying to push this stuff eventually into clinics, then we've got to worry about going to short, stable time sequences in which we don't have the luxury of scanning lots and lots of subjects for long periods of time, which is one way of getting rid of noise effects. So we have to worry about these sorts of noise effects. This is what a good scanner looks like, the Siemens. So this is the min, average, and max in the X and Y direction, no slices outside threshold, and as you would hope, there are no outliers. And here you can see just 0.02%. That's what the average residual looks like. So we can now go ahead and say, well, how does the ACF compare to the original 4th half max measures from FBurn? Because they're measuring it using a different procedure. And it turns out that their measure is per TR, whereas our measure is per slice. But we can go ahead and compare them. So let's look at the FBurn, the minimum 4th half max from the FBurn results against the autocorrelation function measure. So that was when we got the next surprise. And that surprise is that virtually all the autocorrelation measures and lots of the scanners are smaller than the FBurn. In fact, they're ridiculously small. So these are Siemens down here. I can't actually read that here. It's 2 point something, about 2.8, I think, here. This is 1.6. Something's making the autocorrelation function measure a really low value, and it happens up in the GEs and the Philips up at the top there as well. And then if we look at the max, we see exactly the opposite. So the ACF per slice finds lots of examples where it's moved a long way away from the original FBurn measure, and that's basically because you have all these individual slices that are going out of bounds, and the FBurn measure is doing it per TR. So it's tending to average out those individual slice effects unless most of the slices in a given TR are bad. And what do these look like? That one we've already talked about. It's actually one of the green dots up at the top. This one we only discovered a couple of weeks ago. All these guys here that are very low are these high-frequency spatial striping patterns. This is actually from a nominally well-behaving Siemens scanner, our own at Baycrest. So we're in the process now of trying to work out how often that happens and is it a big deal. So we now have both low-frequency non-uniformity and high-frequency spatial non-uniformity effects which show up in resolution measures, particularly per slice. And you can look at the Y direction and you get exactly the same phenomena. Lots of unusually low values for the autocorrelation per slice relative to the FBurn, and lots of large values relative to the FBurn. Okay, so that was the bad news. And I don't know how important this is, okay? I'm going to continue pursuing it because we have seen 2 examples now over the last 2 years where eventually head coils had to be replaced. And when we went back and looked at our measures, we could see things were starting to just very subtly go out of skew in certain slices 9 months before the head coils were replaced. So if you happen to be starting a major study on a scanner whose head coil has decided that it's going to go on vacation, you know, next December, you might want to be tracking some of this stuff, although I don't know how important it is, because if it's only, you know, 100 slices in 5,000, if they're randomly distributed, the chances are it probably doesn't matter that much. If it's 1,000 slices in 5,000, then maybe it matters a lot. So you would hope that we'd be able to get rid of it using some of our pre-processing procedures. This is a sort of moderately bad scanner, one in which the head coil eventually had to be replaced some months later. You can see you've got these big clusters of slices across multiple TRs that seem to sort of go out, and then it comes back for a while, and then it goes out again, this sort of random instability. And you're probably not worried about this, but it occurred to us that we shouldn't just average all these together, so we do a PCA just to check that the pattern is in fact the dominant pattern across all of the outliers. And this is the first component, it's 61%, so that's pretty much the dominant pattern. So the instability on a given scanner in a given session seems to be fairly coherent spatially, but you don't know when it's going to turn up in which TR and in which slices. If we run just a basic pre-processing pipeline with motion correction, censoring, and detrending, so really basic, I haven't even thrown, it was a single scanner, so I haven't thrown resolution effects in here yet. we can get rid of some of these, so that's some good news. It may be, and this is what we're focusing on now, that we want to go to slice-based censoring. So I don't know how many people here have been following, since the Powers paper in 2012, looking at the effects of what we thought were abrupt head motion, but the Phantom definitely doesn't have abrupt phantom motion, I don't think, not across all multiple scanners. So there's two confounded effects there in real human data. There's whatever the scanner happens to be doing, and there's whatever the head is doing in the motion side. And it's maybe censoring adapted to focus on a slice-based effect can actually help us here. So that's what we are currently looking at working on. And this was a TR-based censoring, which seemed to be doing a not terrible job in this particular example. So what have I told you? I've told you I've asked what is the contribution between session and within center scanner effects, and told you that in some scanners it looks as though it might be a problem in some circumstances. And so it should be tracked somehow. And by the way, the standard physics process that happens in most centers is not tracking this stuff. You probably need an F-burn phantom, and then you probably want to do a resolution measure per slice. And I would recommend autocorrelation functions, but you know, there are other ways to do it. And between center scanner effects, well, in our multi-center studies, if you have a scanner that's in bad shape for a year and then they replace the head coil, and you now want to combine all of the Alzheimer's disease resting state studies you did at that site with another place where there's a really well-behaving scanner, that may be a bit of an issue. Because often you end up with very uneven sample sizes at sites. So a really bad case would be you got the majority of your data at the bad site. So I think it is something we do need to worry about. I'm hopeful that we'll be able to remove a fair bit of it by modifying our current pre-processing approaches, using the current AFNI resolution adjustment, but that's a work in progress. And so I'd just like to finish by acknowledging the people that actually did the bulk of the work. So the bulk of it was done by Aditi Kemparathi on the left there, who's a research assistant in my lab. Steve Arnott, who is the operations manager for the neuroimaging in the OBI programs, and Fan Dong, who got it all up and running on a high-performance computing environment so we could run lots of these pipelines over and over and again as we tweak things that we're working on. And of course, it's a multi-center study, so this is, 15 centers, this is, 6 centers. There's the whole team of people funded by OBI at BrainCode, Integrated Data Organizing Center, and Rob Barther is the physicist, and Andre, who's the PI of neuroimaging with Sandra Black, and he put together the acquisition sequences and sort of tweaked them at each site. And Chris Scott at Sunnybrook, some of you may know, has been incredibly helpful the whole way along. That's it. Thanks. I think we need to go to Montreal now. I hope you're not too depressed. Please remember, I don't know how important it is. Well, I'm only at the point at the moment where I know it exists. Yeah, but let's be optimistic. Okay, there's lots of papers which suggest that you can do interesting stuff with existing resting state scans. So clearly this is not a pervasive problem affecting every data set that's out there. Worry